Okay, today we're going to talk about bacterial operons. So this whole lecture focuses on genetic regulation. So what do I mean by genetic regulation? Talked about it multiple times. What do I mean by that, like specifically? What is regulating what? What's being regulated? What?
Genes.
Yes. And what does it mean for a gene to be on?
It means it's
Transcribed. Yes. Okay. So we're talking about turning genes on and off. Okay. Controlling genes, like the switches of genes. Okay. Why even regulate at all? So in the context of eukaryotes, we talked about how there's many different cell types, and different cell types is just a programming of what repertoire of genes are on at a given time. Okay. That's one reason to have regulation. Why else regulate genes? Why not just have all genes on all at the same time? Permanently.
Save energy.
Yes. Okay, that's the big thing is you don't want to be transcribing genes that are useless at the time. And in bacteria, many bacteria have very versatile genomes where a gene might only be pertinent under some context. So they want to have very rigorous systems to turn on and off genes. You only want to be transcribing them when you need that protein. Okay? So that's all built into the regulation. Okay, so we're going to be talking about regulation of transcription. There's different levels of regulation. So there's regulation on / off. So there's regulation like before transcription, on off, like at the promoter. There's the transcript itself, transcript like stability. Okay, and all transcripts have kind of like a half-life. They're not going to survive forever. And then there's also post-transcriptional regulation. What's that? So post-transcriptional would be after transcription, and there's actually post-translational. So there's also regulation that can happen at the protein level, so like stability of the protein. Okay, so just know that there's different, like, when's regulation happening? There's multiple different stages. Before transcription, the stability of the messenger RNA, and then after transcription and translation, there's regulation happening at all these different levels. Okay. So regulation can be pretty complex. So a couple terms you should know. So the enzyme that does transcription is what?
Polymerase.
What kind of polymerase?
RNA.
Yeah, so it's a DNA-dependent RNA polymerase. So you have RNA pol. Okay. So there's other proteins that can bind RNA polymerase and modify its activity. Okay. So one of these is called a sigma factor. So a sigma factor is a protein that can bind to RNA polymerase. And what the sigma factor does is it encodes a preference for the promoters that the RNA polymerase likes. So this is very common in viruses. A certain virus will have a special sigma factor, and that sigma factor can bind to an RNA polymerase and cause that RNA polymerase to find viral promoters and to transcribe those viral promoters. So a sigma factor is controlling like preference of the RNA polymerase. So that's one mechanism of regulating. Okay. You can also have what's called small RNAs. So sometimes you'll see small RNA, sometimes you'll see small interfering RNA, sometimes you'll see antisense RNA. Okay. So if this is a gene promoter, RNA polymerase comes on, makes a transcript. Sometimes there's situations where you can have an RNA polymerase read the other way, and you can produce a transcript going the other way. And if you have a transcript that matches this, but going the other way, what's going to happen with these two transcripts? Yeah. So even though it's RNA, RNA can form double-stranded. So it can form dsRNA, double-stranded RNA. If these sites match, they can anneal to each other. So the point is that sometimes you can get these antisense small RNAs, and they can interact with a messenger RNA. And once that interaction happens, different stuff can happen. That might cause this transcript to become unstable, or it might cause this transcript to adopt a conformation that can't be translated by the ribosome, etc. So you can get regulation based on small RNAs binding to a larger messenger RNA. Just a stat, these small antisense RNAs are 2.4% of the global RNA population in bacteria. I attached the paper that this comes from. So it's a decent amount. It's a decent percentage of RNA that are these weird like regulatory RNAs. Okay. So there's also what's called a riboswitch. A riboswitch is when you have a messenger RNA, and messenger RNA can form more complicated structures. So right now I'm just sort of drawing these squigglies as messenger RNAs, but they can form complicated like stems and loops, things that look like this, okay? And some of these structures can bind to ions, can bind to ligands, some molecule X can bind to the transcript, okay? And that can cause the transcript to be more or less likely to be translated. That's a riboswitch. Okay. And then the final, well, not the final, but another regulatory factor, which I already discussed, which you should know, are transcription factors. And transcription factors, physically, what do they actually do? We'll talk about some specific ones today. What do transcription factors do? Talk to me in words of molecular biology.
They can help locate promoter regions and get it started.
Yeah, I wouldn't use the word locate. I would say they bind promoter regions, and sometimes they're recruiting RNA polymerase, sometimes they're blocking, okay? So there could be a transcription factor that could bind the promoter, and that transcription factor could hold the gene off. In that case, you would call that transcription factor a repressor. It's repressing transcription, okay? There's other transcription factors that will not block RNA polymerase, but will recruit RNA polymerase. They would be like an activator. Okay, so there's different types of transcription factors. They do different things, but all this information, this is like the switch of the gene, turn the gene on and off. And there's many different ways to regulate those genes. Okay, so let's talk about operons. So this comes in the context of, last lecture we talked about eukaryotic genes have lots of introns and exons. They have lots of non-coding DNA. They're very sparse. There's lots of space between them. And then we talked about, in contrast, bacterial genomes are streamlined because there's selective pressure to decrease the length of the code because it's faster to reproduce if it's shorter, okay? So bacteria tend to like cram their genes all together. So they produce genetic circuits that look like this, okay? Where you might have gene A, gene B, gene C, gene D, and there might be a promoter controlling all these genes, okay? This is, the term for this is an operon, okay? So an operon is a series of genes that are coding a sort of like a program. It's like running a subprogram of the organism. Okay, so operons have what I call like 4 characteristics. This would be on test. So one of these is called Synteny. Who knows what Synteny is? Who had genetics? Who's had genetics? Did they teach you a Synteny? They never used that word?
I don't remember.
Really?
They might have, but...
Okay, good. I'm always feel justified that my class is teaching something hopefully useful. Okay, so Synteny is the order of genes. And what Synteny means is that the order of genes is conserved. Okay, so let's say this was a key operon. Let's say this was a key function a key genetic circuit that was regulating some key cell program that all bacteria have, okay? If you look in bacteria 1, let's say you look in E. coli, and you find A, B, C, D in the chromosome, when I annotate chromosomes, what I'll do is I'll draw a line. This means like continues on, and this means like continues on. So here's the operon A, B, C, D in chromosome in E. coli. If you were to look at a different bacteria, let's say Bacillus subtilis, okay? If you were to look for this same operon, because many bacteria share the core conserved genes that are important for life, right? So if you were to find this operon in Bacillus subtilis, it would have the same order. It would have A, B, C, and D, okay? So if the order of the genes is conserved, that means the synteny is conserved, okay? So if you saw this, B, A, C. So in this case, the order is shuffled. You would say this is non-syntenic. Non-syntenic, the order is not conserved, okay? So that's synteny. So one property of operons is that they're syntenic units, where the operon itself is sort of selected to be in this order. And if you look at other bacteria, you'll usually find that same operon in the same order because it's encoding some kind of function to be in that order, okay? That's synteny. Okay, the next one is co-regulation. So usually operons typically are governed by a single promoter, okay? So the genes that are within the operon are all controlled within one regulatory unit, one promoter. That would be co-regulation. So A, B, C, and D are co-regulated by this promoter, okay? Next one is Polycistronic Transcription. Okay, so polycistronic transcription, this has a very specific meaning, okay? What happens in operons is the RNA polymerase will come onto the promoter and it will create a polycistron, which is when the messenger RNA is transcribed, there's no terminator until after the operon, okay? So all these genes, A, B, C, and D, when they get transcribed, they're transcribed as one long messenger RNA with multiple components included inside of it, okay? So then when the ribosome translates this, the ribosome will pick it up here and it'll start translation, and it can translate separate proteins. So if it reads from here to here, it'll see a stop codon there, and it will pop out the A protein, and then it'll keep going. It'll see a start codon and a stop codon, and it'll pop out a B protein. And then it'll keep going, start, stop, C, A, and D. So with a polycistronic messenger RNA, you can produce 4 or however many proteins from one messenger RNA molecule, okay? That's polycistronic transcription. And then the final characteristic of operons is shared function. So that's what I mean by usually this operon is running some kind of a subprogram, a subroutine, and usually all these genes are somehow linked together, related, with some key function that they're trying to achieve, okay? So the proteins work in concert often to get some kind of task done. That's what I mean by shared function. Other things about operons before I go on to specific things. Okay, so operons can be complicated. This is just sort of like a simple basal diagram. You might have operons with multiple promoter elements. You can even have operons with weird regulatory like antisense promoters. So you can have a normal promoter, some kind of an antisense regulatory promoter. Different things can get put into these. So you can have ribosomal binding sites inside of these encoded in. So imagine if you had a ribosomal binding site there, you could encode a sequence, perhaps, that a ribosome would bind here at the beginning, but a ribosome would also bind here. And so maybe your ratios would be skewed. Maybe you'd be producing 2x C protein, 2x D protein. Because every time this ribosome pops on, it goes through the whole thing. Every time this ribosome pops on, it goes to the end. So you're getting 2x C, 2x D, but 1x A, 1x B. So here's an example of how you can manipulate like the genetic code in the DNA to get different ratios of A's, B's, C's, and D's. So the ratio that's being produced is not always 1-to-1, although sometimes it is 1-to-1. Anything else? Sometimes, this is common in viruses, sometimes operons have a read-through terminator, where sometimes the terminator is not 100% efficient, okay? And so sometimes when the transcript is being made, if you have some kind of a weird read-through terminator, there's a proportional probability at which the RNA polymerase will just keep going. And sometimes there's information encoded over here. And so you can encode more information after a read-through terminator, where under some certain condition or some probability, you get other stuff encoded after. So just keep in mind that like regulation, again, can be real complex. This is just sort of like the basics of simple operons. Okay, so let's do an example of the most well-studied operon in biology, which is, who knows what it is? The Lac. Did they teach you that in genetics? Yeah, okay, so they did teach you that in genetics. So we'll review that. Okay, so the Lac Operon. So I have a picture of it. So let's draw the Lac Operon. There's a promoter. I can never tell if this is Lac I or Lac L. Somebody should look it up for me and fact check me. Lac Z, Lac Y, Lac A, terminator. Okay, so here is one of the most simple operons, one of the most well-studied operons in bacterial genetics. Okay, what does the Lac Operon do? What's the subroutine, the subprogram? What do you think it is? So Lac stands for what?
Lactase?
Lactose. So Lac stands for lactose. What's lactose?
Sugar?
It's a sugar in milk, okay? So you've got some operon that has to deal with sugar. What do you do with sugar, usually, or sometimes? I suppose you can do a lot of different things with sugar. What do bacteria like to do with sugar?
Eat it.
Yeah, they eat it, okay? So lactose is actually kind of like a rare sugar, right? It's like a milk sugar. So this operon is regulated to be useful when the bacteria sporadically encounters lactose, okay? And when it encounters it, it wants to be able to eat it and digest it and take nutrients from it, okay? But you don't always want the Lac Operon on because you're not always gonna be exposed to the lactose sugar, okay? So briefly, to go through some of these genes that are included in these, you don't need to memorize what they are, but this is beta-galactosidase. This is an enzyme that's working on lactose. One of these is a transporter. I don't know which one is a transporter, just know that one of these is a transporter. What do I mean by that? I think it's Y. I think it's Lac Y is a transporter. What's a transporter? Anybody know what a transporter is? Okay, so here's a cell. Where is the lactose going to be? Outside. So what do you need to do to get to eat lactose?
Bring it in.
You got to bring it in. Like there's a cell wall, there's a membrane. The lactose does not just like diffuse through the cell, okay? So you actually need to encode a protein, which is, and I did check, it is Lac Y. Lac Y is a transporter, okay? So when Lac Y gets made, it makes a protein that goes into the membrane, and when Lac Y protein sees lactose, it transports it into the cell, okay? So if you don't encode the transporter, you're not gonna be able to eat lactose. Okay, so it encodes beta-galactosidase. That's what's like enzymatically working on the lactose sugar. It encodes a transporter. What is A? Some kind of a galactose-acetyltransferase. So these two are essentially like in the metabolic pathway, and this is the transporter. So this is sufficient. These genes are sufficient to be able to digest and utilize energy from galactose. Okay, and these three are inside of the operon. There's another gene. Let's say it's Lac I, stands for Lac inhibitor. What do you think Lac inhibitor is?
Is it a repressor of the gene?
Yeah, so that's the regulator. So this is a common pattern that you'll see with operons is you might have the operon and then right next door, you'll have encoding of the regulator, okay? So this is the transcription factor. And guess where the transcription factor binds?
To the promoter?
To the promoter that's in front of the Lac Operon, okay? So in its native state, the Lac Operon is off, and it's off because Lac L gets transcribed, or Lac I gets transcribed, made into a protein, which comes and sits on the promoter, okay? And this blocks RNA polymerase. So it's a repressor, it's the Lac repressor, okay? Guess what turns on the operon?
Presence of lactose?
Yes, okay, so you add lactose. Okay, so here's a sugar, lactose. And remember that each of these proteins is a structure of amino acids, and each of these amino acids has different R groups, and it's forming a conformational shape, and that shape of this transcription factor is perfectly evolved to grab onto lactose, okay? So when lactose comes and appears in the cell, this transcription factor has a much more preferential binding to the lactose than it does the DNA that it's binding to. So what happens is the lactose will find the transcription factor, it'll bind it, and then it will fall off the DNA, okay, releasing the DNA. So in the presence of lactose, this thing now turns on, okay, and now you can get transcription of these genes to pump in more lactose and to digest the lactose that's in the cell, okay? So this is an entire subprogram of 4 genes built to detect lactose and turn on the operon if lactose is present so that it can eat it. Are there any questions about that? Okay, so on the test, I ask things, I call this kind of like the circuit logic. So there's some operons I will need you to memorize. This one you need to know. This one's very important. So the logic structure of this is just you add lactose and then the gene turns on, okay? Realistically, in biotechnology, we do not use lactose. We use a different sugar analog called IPTG, which is an analog of lactose. Does anybody have a guess as to why we would not use lactose in an engineer? So maybe I should give more context. In synthetic biology, oftentimes what we want to do is we want to capture regulatory elements that we understand and program them to be able to be manipulatable under an experimental context. So one of the reasons why you have to memorize the Lac Operon, and one of the reasons why it's so important, is because many of the plasmids, which we'll talk about, I think, next lecture, many of the plasmids that you work with, where you put in your gene X that you want to study, oftentimes they have the Lac promoter. Okay, so if they have the Lac promoter, and on the plasmid they have the Lac inhibitor, that means in a native state, your gene X, when you clone it in there into that plasmid, is off. And then to get your gene to turn on, what do you have to add?
Lactose.
So if you add, so you see now you just encoded a switch in front of your gene X, where if you want your gene X to turn on, all you need to do is add lactose. And what I was about to say was, in the lab, we don't actually add lactose. Nobody uses lactose. We add an analog, which is called IPTG, which is similar to lactose, but not the same. Does anybody know why? What's gonna happen in this setup? What's gonna happen if you add lactose to the cell?
It's gonna get eaten.
Yeah, this bacteria, this is just a plasmid, but this bacteria somewhere on its genome has the Lac Operon okay, in its genome. And if you add lactose, it's just going to turn on this operon as well, and it'll eat the lactose, it'll go away, and then all of a sudden your gene will be back off again. So if you want your gene to be perpetually on, you add this sugar, which is the analog, which is called IPTG. And the reason that you use this instead of lactose is because the cell can't eat this. So you add this, it takes the transcription factor off, it turns your gene on, and now your gene is permanently on because that IPTG cannot be digested. Does that make sense? Okay, so now you're starting to understand why the genetics of operons are important. It's because in synthetic biology, we're taking these regulatory elements that we have a deep understanding of, and we're building them into our genetic structures so that we can control the on-off of our switches. Anything else? Okay, so let's do the next more complicated one. So Lac Operon is still used very commonly, so you need to know that. Okay, let's talk about the Ara Operon. The Ara Operon. What do you think the Ara Operon codes for? So arabinose. Arabinose is another sugar. Maybe you're seeing a pattern. What do you think the subroutine of the Ara Operon is?
A transporter and enzymatic components.
Yeah, I mean, this is essentially just now an operon that the subroutine is to eat arabinose. Okay, so now you're sensing, like, again, like every organism is a set of genes, and those genes do specific things. And so now you can see that, like, within the subroutines of bacteria is this little program that allows it to eat arabinose. Arabinose, lactose, every kind of like metabolite or sugar probably has a little subroutine that allows the bacteria to eat it. So this is actually like the method, if you think about it in terms of like computer programming. Okay, so I'll try to like simplify this. So in this case, it's a very similar setup, but what's happening is different. Okay, so in this case, which of these do you think is the regulator? It's weird because there's actually two genes with the same name. Let's call this 1, 2. Which gene do you think is the regulator?
Ara C.
Which,
one.
yeah, this one. So this is the regulator because it's kind of sitting like outside, okay? So this encodes the transcription factor, Ara C. Okay, and in its native state, if I would say expand this, so DNA is kind of like long, right? So if you were to extend this into the genome, imagine a situation that looked like this, where Ara C binds to 2 DNA sites, one further upstream, like over here, and one close here, and that causes a loop in the DNA, okay? And that loop in the DNA blocks the transcription of RNA polymerase. So right now, in its native state, there's a stem loop in the DNA that's blocking RNA polymerase from transcribing these genes, okay? Now, if you add, guess what you add?
Arabinose.
Arabinose. Okay, so the logic is you add arabinose, and the gene turns on. Okay, so in this case, you add arabinose. Do that with blue. When you add arabinose, it binds to Ara C, and there's like a conformational change, and now the preferences, the DNA preference sites that this thing prefers are subtly different. And it shifts to grab this like other site up here, releasing the stem loop, okay? And now it's, in this case, it's not blocking, okay? So this is, it's not blocking the RNA polymerase under this conformation. And then the RNA polymerase can come on and transcribe, and now you get induction of B, A, and C. Okay.
So is it the same thing where the arabinose attaches to Ara C1 and then it falls off of there?
So that's it. So that's what I'm trying to clarify. In the Lac Operon, lactose is binding to the transcription factor and it falls off. That's not what's happening here. What's happening here, it's similar. What's happening here is that arabinose is binding to the transcription factor. There's a conformational change in the protein. It then prefers to grab a different DNA site, which is closer to the promoter. So it's essentially like, in one case, in the Lac case, it falls off. In this case, it doesn't fall off, but it releases the stem loop.
Okay.
Does that make sense? Like, that's the simplest way to say it. So what you're understanding is transcription factors can mechanistically do different things, like that can cause on or off states. So in this case, it's not falling off the DNA. It's actually still there. It's just releasing the stem loop. Does that make sense?
Yeah, that makes sense.
Okay. How much time do I have left?
19 minutes.
19 minutes, plenty. Okay, so these are the only two operons I will make you memorize for the test. There's another one, trip operon. I won't make you memorize that one. These ones I make you memorize because we use these in the lab. These are super common. Okay, so you kind of want to know how they work. Pros and cons. The pros and cons. The cons of the Lac system. The Lac system is known to be leaky. I'll talk about this more later. What do you think I mean by leaky? So its natural state is that repressor is bound and it should be off. What would it mean if it was leaky?
Does the repressor fall off?
The repressor stays on, but sometimes the RNA polymerase can kind of like squeeze by. So you get kind of like leaky transcription. So there's like another thing you'll learn is, so there's various states of off. Like there's a real tight off. The Ara Operon is known to be a very tight off, okay? So Ara is a tight off, okay? The Lac Operon is not a tight off, it's a leaky off. And actually nowadays nobody even uses the basic Lac Operon, they use an engineered Lac Operon, which we'll talk about later, okay? So cons of the Lac is it's a little bit leaky. It allows us just a little bit of transcription, okay? Another pro of the arabinose system is arabinose is cheaper than lactose. So arabinose is cheap. And the arabinose system is also, it's titratable. What does that mean? Titration in chemistry means what?
Reaching equilibrium, like you're trying to get to equilibrium.
That probably kind of makes sense in some sense. What titration means in this context is that the more arabinose you add, the more transcription you will get. So if you have like 1, 2, or 3, you'll get 1x, 2x, 3x. That's a titratable system where you can increase the arabinose and get more and more production of transcription. The Lac Operon is not so titratable. It's kind of more or less like if it's there, it's either on or it's off. So it's not as quantifiably titratable, okay? So if you had a situation where you had a plasmid, where you had gene X, and you had the Ara Operon controlling your gene X, in this case, you could actually do experiments and you could figure out, well, I want exactly this much gene X, therefore I should add exactly this much arabinose. And you can figure out roughly, like proportionally. You can do that to some extent with Lac, but not as much as the arabinose system. So that's one advantage of it. Other advantages. So another advantage of the arabinose system is that if you add, I'm kind of piling out a lot of stuff. If you add glucose, it actually represses. So the arabinose system, you can turn it on with arabinose and you can shut it even further off with glucose. So there's like multiple things you can add that adds the complexity of the switch that you can control. Any questions about that? Okay. Let's see. That was quick. That was easy. Let's do some more experimental questions. Okay, so last time the question was detect the presence or absence of a microbe in a bug. Remember that, and the answer was PCR. Okay, in this case, let's do a variation of that question. So this would be experiment 2, detect presence or absence of microbe X in a bug, but in this case, there's no genome for the microbe. So in the last case, we talked about, like, maybe the example was detecting Wolbachia. And what would the genome facilitate in that case? Like, how were you able to use a detection method to detect it based off a genome?
Primers?
Yeah, so we talked last time, the answer was, well, you can do PCR and you can run a gel. And the reason that you can do PCR is because you can design primers that would match some sequence from some gene, let's say gene X from Wolbachia, okay? And the only reason you can do that is because you can pull the genome of Wolbachia. You can design your primers based off that genome. Well, what if there's no genome? What if you're studying like a new microbe that has never been identified before, and you want to know, like, you have a hypothesis that it's there, but you don't know if it's there? How could you develop a strategy to detect it? Let's say you kind of have a hint about what it is, but you don't have a genome. What would you do?
Could you make antibodies?
That's not the easiest thing. Like, I want to teach you guys like the easiest way. So I'll give you hints. Okay, so you can still do a PCR. Like, you could do a PCR. You just have to think about it more carefully. How would you optimize a PCR if there was no genome? How would you design the primers? What would you do?
Well, we know it probably has the Lac Operon, or
That's a good idea. So you're getting a good idea. I would not target the Lac Operon, but your idea, if I can rephrase it, and you tell me if I'm right, you're saying there's some genes that all bacteria have. So therefore, if there is a bacteria in the sample, we should be able to target a gene that all bacteria have and see if it's there. Is that your rationale? Yes, that's correct. Okay. So you would PCR based off that's the term for that is a conserved gene. I would not target the Lac Operon because actually a lot of bacteria probably don't have the Lac Operon. But what's something that you might target? So then you bring up these interesting questions like, okay, well, which gene should we choose? What gene might be good for this?
a gene that codes for like pili or something.
That might be good. Although, like, stereotypically, there's something that kind of like people just always choose.
Could it be like based on if it's gram negative or gram positive?
You kind of want a situation where whether it was gram positive or not, it still work. So in most of these cases, people choose ribosomal genes. So they'll choose like R DNA or R ribosomal. I forget the specific one, but there's a ribosomal gene that it's 16S. So you've ever heard of 16S sequencing? So that's a ribosomal subunit, 16S, that they are sequencing the gene from, okay? And the nice thing about that gene is there's regions of that gene, so this is the whole gene. There's regions of that gene that are conserved, meaning they never change, and there's other regions where you can acquire a few like SNPs where you get some changes, okay? So if you do a PCR on this, you can design your primers in the conserved regions, and then you can PCR over an amplicon that covers the variation, and then you can sequence that and you can figure out like roughly like what it is, okay? So that's how you would detect the presence or absence of microbe X in a bug with no genome, okay? And probably what you would want to do is if you had a hypothesis for what you think it might be, you'd probably want to construct what's called an alignment. What's an alignment? So if all bacteria have this gene, 16S, you could pull sequences of 16S from many different bacteria, let's say here's 6, and you line them up and you see where they match. So if they match you'll get like hits, and where they don't match, you'll get like blanks, okay? And then you'd want to design your primers in regions where they're conserved. So that's how you would actually like find the spots. To find the spots where they're conserved, you'd have to construct an alignment. You'd see where they're all the same, and then you would design your primers in that little region. Does that make sense? Yeah?
So what would happen if you ran 16S and you got, is it possible to get multiple back? Like, oh, this bug has multiple kinds of microbes?
Yeah, okay, so that's an interesting question. Okay, so good, I like this. Okay, that actually makes it fun to teach. So if you had a situation where you had multiple bacteria, right? What might happen is you'd run your PCR and you'd get bands. They'd probably be all the same size because the gene is probably gonna be the same size. It's just that the sequence is different. So you'd run a gel. You'd only see one band, but that could be representative of multiple different sequences in there. So how would you even know if there were multiple sequences? What would you have to do? What technique? So your first step would be PCR. Second step is you run your gel. You get your band. Then what do you do?
Is it SDS page?
No, SGS page is protein. This is nucleic acids.
Oh, like, I meant like with like bacteria.
Nope.
We don't know.
What would you do with this? So the gel's only telling you the size. So let's say it's 200 base pairs. To answer your question, you want to see the sequence. So how do you get the sequence? So I talked about different ways to get sequencing data. Which one would you pick? What are the two options? I only talked about two.
Next gen and Sanger.
Yeah, so there's Sanger and there's next generation. Which one would you pick and why in this case? It's a clear answer.
Illumina, high fidelity sequencing next gen.
Why?
Because you have
It's 200 base
multiple.
pairs.
No, so I told you, in this case, you only have one thing. It's an amplicon of 16S PCR DNA. That's all it is, it's one thing. You might have some variation within that sample, but you don't have like a billion different sequences you can see. So you're only gonna use next generation if you're sequencing like genomes. Okay, so think that in your head that you're never gonna use next gen. I mean, there are situations, okay, so now I'm like shooting myself in the foot. There are situations where people will do like next generation sequencing for 16S to get a whole bunch of stuff real quick. In this case though, where you have a PCR band, it's just one thing. So you just do Sanger sequencing. And what you'd find is in the chromatogram, you'd get the chromatogram, and if there were multiple things in the sample, what would you see? At certain positions, you'd see reads with conflicting bases. So maybe you'd have like a T or an A, or a T and a G. This would indicate that there's probably like multiple things in that sample. Does that make sense?
Yes.
If you had, here's the one case where you would use the next generation sequencing. If you had like a sample of like salt water from the ocean, and you're like, I think there's probably like maybe 1,000 different microbes in this sample, then you might use next generation sequencing to sort that out. Does that kind of clarify? Okay, good question. How much time do we have?
6 minutes.
Okay, let's do another one. Let's see. Okay. So, this is good because this would be good for the quiz. So, we'll talk more about plasmids, okay, next lecture, I think. But oftentimes what you do with your plasmids is, plasmids are a frame to build on. So oftentimes you're doing like an insert, where you're cloning a product or gene that you want to study, and you're putting it into a plasmid, okay? So a common thing that you want to do with plasmids, here's experiment 3, is you want to check to see if plasmid X has your insert, which is gene X. Okay, so I'll give them different names. I'll say plasmid Z. Okay, so what technique could you use to know if plasmid Z has insert gene X? You tried to build this, you want to know now were you successful? Did you successfully build it? What would you do?
Insert an antibiotic resistant gene and plate it.
So all plasmids by default we're jumping ahead a little bit, but all plasmids by default are going to have some antibiotic resistance cassette, like AMP. So whether the plasmid has an insert or not, it's still going to be antibiotic resistant.
transform equal lines?
What?
Transform?
That would be in part of the process. You'd have to transform it for sure. But the question is, how would you look at the DNA and see if it's there?
Could you use I can't remember what the jellyfish.
So that would mean you would have to re-engineer it. So you could like add a tag. You could add like a GFP or a fluorophore tag, and then you could somehow like see that it was green. But that's kind of like, that's a lot of like extra work. Is there any way to just see whether or not? So what did you start with? You started with this, and you got this. Is there any way to tell the difference between this versus this?
You run a gel?
You run an agarose gel. So again, people think like way too complicated. The only technique that you need is you need a technique that can tell you the difference in size, which is an agarose gel. So you run an agarose gel, okay? So your lane 1 will be your ladder. How big are plasmids?
Small.
How small is small? How many base pairs? We haven't had the plasmid lecture yet, so I forgive. But plasmids are about 3,000 to 10,000 base pairs in size. Okay, so you have to strategically pick the right agarose gel. So you wouldn't run a 2.5, you'd run 0.8, which means the holes are a little bit bigger, which allows a little bit more bigger stuff to flow through the gel. Okay, and you'd have your 1 kb ladder, so you'd have 1,000 base pairs, 2,000, 3, 4, 5, 6, etc. Okay, and what would be your negative control?
One that you know you didn't list.
Z. So the one you started with would be your negative control. You got to keep a little bit of that. Okay, your experiment would be this. Okay, so you'd be comparing these two lanes. What would you actually see? So you purify this plasmid. What would you see on the gel? And this is actually a question from the quiz. What would you see on the gel if you run blank Z plasmid? What would you see?
Since you're allowing bigger stuff to go through, it would be like all the way.
So what should I draw? What would I see?
A lot of banding.
A ton of banding. Okay, so if you're saying I would see something like this, what would this suggest?
Small.
Huh?
It's a small band.
If I saw this, what does this mean?
An unpurified sample?
It could mean that it's unpurified, or it could mean that Z was like chopped up into 1,000 different pieces. And hopefully you don't see that. What should you realistically see for certain? If you have a purified sample of Z, what should you see?
One clear band?
Yes, at what position? Let's say Z is equal to 4,000 base pairs.
And beside the pool?
So you should see that, in theory. Okay. You will see that, but you'll also see two other things when you run plasmids. So when you run native plasmids, closed plasmids, which are circles, you'll usually also see a band like this and a band like this. Okay. So I'm telling you now what these bands are. This is a cool, because if you do synthetic biology, you'll work with plasmids for the rest of your life. So this is kind of like an important thing. This band is the closed endogenous like one molecule, okay, where it's running right at the size you would expect it. This band is what's called concatenated plasmids, where sometimes when plasmids get replicated, they can get tangled with other copies of the plasmid. And if they are entangled, they can't break apart, okay? They're covalently sealed like in tangled loops. So this is much bigger than a single plasmid, which is why you usually see a higher band, which is moving slower. So the higher band is the concatenated plasmid. This band on the bottom, which is running faster, is what's called supercoiled. So in this case, imagine you took the plasmid and you grabbed it and you twisted it like a rubber band. You just kept twisting it. Okay, what would happen is you'd build up tension in the plasmid and it would start to coil up and form like a little knot. Okay, so the knots run faster, the supercoiled DNA runs faster. So whenever you run a wild type plasmid, you're going to see 3 plasmid bands. You'll see the concatenated, the normal, and the supercoiled. Okay, so this is what you should see. Now answer the final question. Check to see if plasmid Z has an insert. What would you see? So in your X plasmid, if you got it right, what would you see?
Lower than 4.
So you're saying you would see This?
Yes.
Why is that wrong?
I don't know. Because that means that the DNA got smaller, but yeah.
Yeah, this means that the DNA got smaller.
Oh, okay.
So this would mean that you messed up your sample because it should not get smaller, it should get bigger with the insertion of gene X. So let's say gene X is 1.5 kb. You should see, so it's 1.5. You should see this, this is an upshift of these bands, okay? So this is a very, very simple visual experiment. Anybody can do this. This is the most simple cloning experiment you can possibly do, is checking for sizes of plasmids. And this is usually the first step in confirming that you got your gene inserted in plasmid Z. How much time do we have left? Are we done?
We're done.
Okay. Can I just say one final thing, which is, this tells you the size, but what does it not actually tell you?
Just the right gene.
Yeah, it doesn't actually tell you whether you have gene X. It tells you that you have something inserted that's approximately the right size. So typically you follow this up with saying sequencing, then you know both the size and the sequencing is correct. Okay? Good lecture. You should